Thanks very much. Thanks everyone, and thanks to the organizers for asking me to participate in this session. It's a real pleasure to be here. I've kind of changed the topic of the presentation to kind of map in with hopefully the target audience, which is around pathology, and really to pick up on the last question that was asked around transitioning algorithms into that clinical environment and putting them into the hands of pathologists for routine diagnostic purpose. I do realize I'm standing between everyone and a trip around Coventry or a drink of beer in the bar, so I will be as brief as possible. I think I've only got 140 slides, so we should be okay. I always like to start these lectures with a reminder to everybody that computational pathology, digital pathology is not new. It's been around for a long time with a long history of academic papers and publications and research. I published my first paper in digital and computational pathology in 1987. Yep, 1987. I was 5 years old at the time. Doing my first PhD, but back then I was developing applications that use digital images and algorithms to characterize the subtle changes that appear in nuclei in early stage colorectal cancer. And clearly it's developed hugely over the interim period. But it's been around for a long time, and I've been around for a lot longer. But the key advantages, as we all know, and for pathologists in the audience, you'll be very familiar with this, having attended conferences like this, is the objectivity of computational pathology, the fact that it allows you to convert images into numerical information, which can then be used to support diagnostic classification, which is much more reproducible, and allows you to define diagnostic patterns or morphological patterns with a higher level of precision. It allows you to take what you currently see today when you look down the eyepieces of a microscope or when you look at a digital image. It allows you to measure what's seeable, so measure those visual clues that you use routinely. But it also allows you to detect the unseeable, those characteristics that are subvisual, that escape what you can see with the naked eye, and allow you to characterize disease in ways that have not yet been possible. But the key question that I ask myself a lot, and that we ask ourselves within Philips, is why is computational pathology not aiding pathologists in clinical practice today? So why hasn't made that transition, that leap into routine clinical practice with all of the benefits that comes with it? And I think this comes down, there's many reasons of course, but in my mind it comes down to four key areas. Most of the solutions that are available today have been developed for the research environment, for the researcher who's investigating new ways of doing image analysis. The audience that Pete has already referred to. And to be able to take those types of solutions and use them in routine diagnostic practice requires shifting of context, asking the pathologist to take the images that they're looking at routinely, load them up in a different software platform, begin to apply algorithms with it, and then take that data in a meaningful way and use that for routine diagnostics. And that stepping out of those workflows, stepping out of that routine workflow, and that context shifting is not convenient and doesn't make it easy for pathologists to adopt these technologies. Many of the algorithms require significant interaction, so the pathologist needs to go in and set thresholds for certain samples to be able to define what the algorithm is measuring. And again, for routine diagnostic practice where pathologists are facing significant daily workloads, that effort is often just too troublesome to adopt. Algorithms tend to be quite time-consuming, and not in all settings, but in many settings, where the pathologist has to annotate areas and then sit and wait for the algorithms to run and generate the data. And of course, again, in a very busy routine diagnostic laboratory, that poses challenges. And in many of the applications that have been developed and that I've been involved in and publishing on, lack proper validation. where the pathologists using this for routine diagnosis and where the impact or the output of those algorithms are going to be important for clinical decision making. If they're not validated and the pathologist does not have confidence in them, then clearly it's going to inhibit adoption of computational pathology. I think one big transition that has happened, as we all know, over the last 10 to 15 years, has been the development of whole slide imaging systems and digital pathology workflows. Not just for research, but digital pathology solutions that can be embedded within a routine diagnostic laboratory where high volumes of slides can be scanned routinely and where pathologists are now viewing these images digitally as part of routine diagnostic practice. Obviously, within Philips, we've developed a high-throughput scanning platform and a comprehensive workflow that supports the use of digital pathology for routine diagnosis. And this provides the framework on which we can then begin to layer AI applications into that workflow, making them much more convenient, available to the pathologists in a completely embedded and seamless way to their existing digital workflow. And I think that's going to be the big game changer in making that transition of algorithms into clinical practice. We've already made some starts in this direction, some advances in this direction. We've embedded a range of algorithms for the breast IHC panel, ER, PR, Ki67, HER2, where these algorithms that have been validated on large data sets, have been tested to perform and provide objective quantitative data, but where they're not sitting as a separate application outside of the workflow, but where they're embedded within the digital workflow. So pathologists can view that breast case, the H&E images associated with a difficult breast case, where they can then subsequently view the IHC images, the IHC panels, but select algorithms to run on those images to generate data as part of the routine workflow. And I think this is significant. And these algorithms, as I say, have been validated extensively. They're from Visiopharm, so they're CE IVD cleared, so they've shown to be better, faster, cheaper, and safe. And where they significantly reduce inter and intra-observer variability in the assessment of these markers. But of course, we've been hearing at this conference about the huge advances that are taking place in artificial intelligence and machine learning and deep learning. And clearly we're beginning to see how this could have a significant impact on routine diagnostic practice and really changing how pathology is performed routinely. And within Philips, we are absolutely interested in how this is going to revolutionize and transform pathology. And we now have teams of very gifted and talented data scientists working on these problems within the organization. Some of them are in the audience today. And it's a real privilege to be able to be at the forefront of developing some of these novel applications that we believe, by embedding those within routine diagnostic practice, are going to make a huge difference. Obviously, with deep learning applications, historically there's been a lot of image analysis applications focused very much on IHC, but as we've already heard extensively in this conference, deep learning has huge advantages for interpreting complex images that we see in H&E as well as in IHC. And we've been working on a range of approaches and algorithms that allow you to automatically identify tissue compartments and tissue patterns, to be able to use deep learning for cellular analysis and identification, for using deep learning to perform the IHC measurements, which are so important in many diagnostic decisions. And certainly in our hands, we're beginning to see that deep learning can significantly outperform conventional machine learning or segmentation classical imaging approaches. And of course, deep learning can also be used to not only measure what pathologists routinely assess in terms of biomarkers, not only for segmenting tissue areas, segmenting nuclei, for measuring IHC markers, but also to take that pixel information, to ingest that and generate new signatures of disease that are completely transparent to the naked eye, but which can add significant value when we're thinking about tissue classification, disease classification, prognostication, and prediction of response to therapy. So these are the new tools that we as an organization are actively involved in developing and embedding within our solutions. Sorry. And of course, having access to these types of data that can be generated from images gives us unique ways of visualizing that data that allow us to be able to visualize changes in tissues and cells, to be able to overlay heat maps on top of the tissue so you can see key areas of change, you can see key areas of cancer within the image, to be able to map the cellular locations within the image and look at the cellular sociology, so the relationships between different cells, and to be able to map those in really clever ways that allow pathologists to visualize tissues and visualize samples in ways that have not been happened before, including presentations of the images such as ring formations where you can map the different characteristics of the image, and depending on how many rings are closed, you can determine what the underlying characteristics of that tissue are and determine what the diagnosis is. We call this augmented visualization, but it's providing pathologists with tools that augment their existing visual capacity, but provide them with new tools that they've never had access to before. I want to give you a quick example. I'm very conscious that that as a session, we're getting close to the end of time, but very quickly I want to give you an example of a solution that we developed within Philips to support the automated markup of H&E tumor samples called TissueMark, and it's designed to automatically mark up samples for molecular macrodissection. In molecular labs, where techniques such as next-generation sequencing, molecular profiling, in solid tumors, the starting point for that is absolutely critical to the success of those technologies is pathology, reviewing the H&E tissue sample to determine where in that tissue sample the tumor exists, to annotate that for macrodissection, but also to precisely estimate the percentage of tumor cells within that sample. If you don't have sufficient tumor DNA within the tissue, then the downstream molecular assay that's assessing specific features within that tumor DNA will be flawed. So really important to reliably do this in using histopathology. And in many of the big commercial molecular labs, they're employing armies of pathologists to do nothing else but annotate tissue samples and review the percentage of tumor cells within those. But pathologists, as pathologists know and as we know, are not terribly good at quantitatively assessing the percentage of tumor cells within tissue images. These are just some data that we've collected and that we have from others where we see that the variation both within pathologists and across different laboratories in assessing the percentage of tumor cells, the variation is extensive. In some cases, one pathologist will call it 25%, another pathologist will call it 85%. Huge variation in how they're calling these samples, and that has a significant impact on the downstream reliability of the molecular tests that we're applying routinely in clinical practice. So we've developed a deep learning technology that allows us to automatically identify tumor within H&E tissue samples. Allows us to create an automated macrodissection boundary that can then be used to inform the lab tech in macrodissecting that tissue. And in addition to identifying tumor tissue and and defining an automatic macrodissection boundary, we can also very specifically identify tumor and non-tumor cells within the H&E tissue sample. I think if you saw Manuel's presentation earlier on, you'll have seen this image in his presentation too, where we can use deep learning techniques to identify those cellular populations and specifically identify and distinguish inflammatory cells from normal cells, from tumor cells, in order to provide us with a quantitative measurement of the percentage of tumor cells within these tissue samples, in a reliable, reproducible, and quantitative way that is so difficult to achieve using visual interpretation. And when we do this and compare this with objective standards and objective baselines, we show very high performance of the automated algorithm against pathologists. Now, this isn't pathologists eyeballing it. This is where we've asked pathologists to go into those tissue samples and by hand count the numbers of individual cells within each one of those images. Hugely time-consuming task. It takes up to 16 hours for pathologists to do one slide. The pathologists that we used in the study are still in rehab as a result of that experiment, but it gave us the baseline, the objective baseline to compare our algorithm against. And we've shown that using TissueMark, which can calculate the percentage of tumor cells in less than a minute, provides us with that objective measurement that we need. Of course, outside of TissueMark, the literature has shown the advantages that the deep learning applications and computational pathology can have in many other areas of diagnostic application. We've already talked about breast IHC, but you can see the types of time savings that can be made by using objective automated interpretation of tissue samples, including prostate to tumor detection, and, as we've already heard today, breast sentinel lymph node biopsy evaluation, the detection of metastases within those lymph nodes. And for pathologists in the room who have sat looking through many slides of breast lymph nodes, looking for that very small island of tumor cells, you'll appreciate the real advantage that this can bring in terms of the time savings, which translates into cost. And the real value of these types of tools are where you can take a series of lymph node sections from a specific patient. Those can be scanned and analyzed using deep learning algorithms. And where, it probably doesn't come across terribly well on the screen, but where you can identify those lymph nodes that are suspected of having micro or metastases in them. But the real value of this technology is how you then interpret that data and use that to reorder the cases and select the cases that the pathologist should review first of all. And the pathologist that can then review those cases that are much more likely to contain metastases than others which are detected to be absolutely normal. And this type of approach to using deep learning in routine diagnostic practice will have a significant impact on performance. We've seen over the last year and a half a huge growth in the development of new companies that are actively pursuing deep learning for pathology. Over 100 million, I think it's now 140 million of venture capital investment has gone into companies that are specifically focused on pathology. Massive growth in the interest that this is having. We've seen a huge growth in academic research in this area. The number of papers have grown exponentially in the last number of years that are applying deep learning and AI technologies to pathology. Government is also backing these initiatives. You will have heard in a number of sessions, and you'll hear about it a little bit more tomorrow morning in a dedicated session, about the UK Innovate Initiative. This is where UK government is investing behind digital pathology and AI in pathology to develop new applications that are going to transform the delivery of pathology in the UK. Delighted, obviously, that Philips, the company that I work for, is actively co-investing in these initiatives. And we're working closely with two large clinical hubs, one based out of Scotland, one led by Coventry, with Oxford, Nottingham, and Belfast being partners in that, where we're going to work together with those sites to develop and transform how deep learning and AI can be applied in pathology. 4 key goals for these initiatives: lab digitization. So Philips as an organization will fully digitize these labs so that they have the capacity to move to 100% digital operations, where every single glass slide going through those laboratories will be digitized. And on the back of that digitization program, we're going to create the largest data lake in the UK with a range of images across different diseases and different tissue types that are highly annotated by pathologists in the UK to support AI and algorithm development. We're working on a range of specific algorithms in partnership with those centers. This hasn't been driven by Philips, but these are algorithms that the pathology teams within those centers have selected to be algorithms that will transform their workload and their practice. And we're also taking those algorithms right the way through to validation so that we ensure that they're safe in those clinical environments and can be utilized routinely. This is particularly important in the UK. Recent Royal College of Pathologists survey has shown that only 3% of departments in the UK have enough staff to deal with the routine workload. We need to be thinking about innovative and creative solutions that are going to help overcome the workload, work staff crisis that we have in countries like the UK. And we're seeing this in other parts of the world as well. And AI is going to have a significant role, but only if we can translate those algorithms into clinical practice by embedding them seamlessly within the workflow that pathologists have. We need to do this in a very clever way. Got a few more minutes, hopefully. Obviously, research is extremely important in this space. And many of the publications and presentations at this meeting are hugely inspiring and show what can be done if we're creative and innovative as scientists and pathologists. But they tend to be based on small training sets, very often from one laboratory. Limited pathology input. I've seen so many publications coming from computer science labs that have very little pathology input, and that is clearly a major issue. Very often the algorithms have poorly defined claims, very limited validation, no qualification of the algorithms, and really no health economics evaluation. And there's justification for that because they're being developed in a research environment. But to be able to translate those into clinical practice, we need to think about this in a different way. And this is what Philips, as an industry lead in this space, are spending a lot of their time thinking about to ensure that we can deliver algorithms in a safe and compliant way. We need to develop these on extensive training resources across multiple laboratories. We heard about it earlier. It's really important that these algorithms can generalize to the global audience of pathology labs. And so we need to extend our training resources and the number of labs that are contributing to our data lakes. We need to ensure that we've got consensus evaluation from multiple pathologists so that when we're annotating samples that we've got consensus views so that we can build algorithms that perform highly in clinical settings. We need to ensure as an industry that's delivering these algorithms that we've got really strong product definitions and well-defined claims for the algorithms. We need to have extensive validation of these algorithms across multiple labs. Full clinical studies are necessary if we want to take these from research algorithms into clinical algorithms that are IVD cleared. It's really important that we do those robust clinical studies and health economics evaluation to demonstrate the cost-benefit impact of these. This takes time. This is extremely costly, but there'd be nothing worse than an industry taking an algorithm quickly out into the market where it fails or where there is a misdiagnosis. So it's really important for us as an industry lead in this space to take that responsibility and ensure that we're building these algorithms that are safe and effective in clinical practice. And we've defined a very comprehensive map as to how to do this. That covers a range of different pipelines from algorithm development, the development path, validated tools that are necessary to ensure that we're building robust algorithms, a regulatory path that maps into that entire end-to-end process, and a health economics path as well. These series of activities need to be very carefully orchestrated to be able to deliver IVD-ready algorithms. Where we can take those to the appropriate regulatory authorities, the FDA in Europe, to have them CE-IVD cleared, and to have them cleared by regulatory organizations in different parts of the world. And this is our responsibility as an industry lead in this space to ensure that we're delivering safe and reliable algorithms in clinical practice. These algorithms can be defined in a series of ways. I just want to show this in the last few minutes. this you will recognize as a typical workflow within a clinical pathology, diagnostic pathology laboratory, where you have slide production at one end, they're dispatched to pathologists, the pathologists will review them, they may decide to order extra tests or slides, and then they will make a decision and sign that case out. That workflow is being transformed today. We've already introduced scanning into that workflow where all of those slides can be scanned in a high throughput walk-away fashion. So the slides can be delivered to pathologists in a digital way and they can view and make those diagnostic decisions safely in a clinical diagnostic setting. But we're also introducing analysis into that workflow, where as the slides are being scanned, they're also being analyzed at the same time of scanning. And Philips are working really hard to build the correct and appropriate infrastructure within their technology stack to make sure that this happens seamlessly. That analysis will allow us to do a number of things. It'll allow us to ensure that when the pathologist reviews the slide at the time of review, they'll have access to all of the annotations, heat maps, the measurements, the digital tests to allow them to make those decisions and where those decisions will be better informed. Also allows us to intelligently dispatch or prioritize cases for pathologists in the same way as I discussed earlier on in terms of lymph node biopsies. But it also potentially allows us to identify those cases, those samples, where we need to order extra tests and extra slides. Really streamlining and making that workflow much more efficient, not just with digitization, but with computational pathology. I'm going to skip over this slide in the purposes of time. These, as we begin to discuss these with diagnostic pathologists, there are a number of questions that inevitably come up. Pathologists are really worried that AI is going to replace them. We don't see this happening at all. There will be some, sorry, I should rephrase that. I do see some applications where automation will absolutely be used routinely in diagnosis. But in most complex decision-making processes, we see a very close interaction between the pathologists and the AI algorithms that we're developing. Pathologists have real advantages in being able to integrate data from multiple different sources. They have extensive domain knowledge. They can handle noise in ways that algorithms very often can't. But we know the issues around subjectivity of visual interpretation and unintentional biases. Algorithms, as we know, have precision and reliability. The decisions are data-driven. They can detect things that just can't be seen by the naked eye. And it's really the combination of these complementary skills, the integration of human and machine that I think is going to make a massive difference to our ability to make reliable diagnostic decisions. I like to think of it, as others do, that it's less around AI and more around IA, which is intelligence augmentation. So taking the skills that the pathologists have and augmenting those with new tools that they don't have today. The other big question is it all just hype? So we're at this conference hearing a lot about machine learning and deep learning, but is it going to be something else next year or the year after? Is this all just a fad that we're going through? And we can't think about this in terms of the hype cycle, which you'll all be familiar with, is the Gartner hype cycle, where you see how technologies grow and there's a huge amount of excitement and then they dip into the valley of disillusionment. And you can see here that deep learning networks are really at that peak. So next year, are they going to be in that valley? Well, the one interesting thing is that if you look back over the last 5 years, deep learning and machine learning has retained that position. It hasn't dropped. And I think we're going to see this in pathology. There's a real drive and a real motivation to develop new technologies. The technology is now available in so many consumer devices. I think that will become a consumer technology in pathology in the next few years, given the advances and performance that we're now beginning to see. Manuel talked about this earlier on. This was a paper that we published at the end of last year. Pathology as a discipline has gone through many changes, many advances over the last century or so. And we like to think of this in in 3 revolutions. The first revolution, following on from pathology being established as a morphological discipline that really informed clinical decision-making, the first revolution was really around immunohistochemistry and the value that that brought to pathology and being able to identify disease-related proteins and be able to visualize those and use those for better decision-making. Second revolution was really around understanding the molecular diagnostics of diseases such as cancer, but where pathology continues to be a really important facet in understanding the importance of those molecular changes. The third revolution, I believe, is digital and computational pathology. Obviously, working for Philips, really proud that in 2017, we really led the way in getting the first digital scanner approved in the US for primary diagnosis. And that has really set the scene for allowing us now to develop other technologies, including AI, and to be able to layer that on top and embed that within the workflows that pathologists are now using routinely. I think we've the opportunity, the individuals in this organization, the scientists, the pathologists, together with industry partners, with initiatives such as Innovate UK, we have the opportunity to build a really exciting future for pathology. A future that will inform pathologists, that will transform pathology, but importantly, make a huge difference to the lives of patients that we all are here to serve. Thanks very much.
Thank you very much, Peter, for that talk. I would have one question before I start the questions to the audience. Yeah. In terms of validating, because you also had this in your talks, in terms of validating algorithms for clinical use, do you think it's time already to separate the algorithms from the devices? Because as you said, in 2017, where we have cleared your first device for FDA, this was of course a combination of algorithms and hardware, so software and hardware. And at the moment we are still seeing that both are more or less incorporated and then as a whole solution are validated. Yeah. Do you think it is really the time that we can move on like this, or with the growth of computational pathology and the vast amount of algorithms that are available nowadays, don't you think we have to change this regime a little?
I think it's a really important question, and certainly the direction that I think FDA and other regulatory organizations are going is that we will be disentangling those different technical components within that workflow. However, as you know, with many diagnostic devices, they're very much tied to the underlying platform on which they're created. We all know the differences between the different scanners and the image content that is generated. They all look slightly different. They've all got slightly different, they use different optics. So I think in the first wave, there will be the requirement from the regulatory organizations to tie those algorithms into specific platforms. Moving forward, however, clearly we need to look for clever ways of taking those algorithms and being able to tune them to other environments so that we can extend the use of those across multiple different imaging platforms. But from a regulatory point of view, what we're seeing today is that they need to be tied to a specific underlying imaging platform.